We can see sometimes changes that are inherited from a parent in an autosomal dominant pattern, or a new change in an affected child that affects one of the two copies of a gene on a chromosome, which would also look like autosomal dominant, but is a new mutation and not inherited from the parent. We can think about autosomal recessive. Most individuals with autism would fit into what we call multifactorial inheritance. Next slide. In multifactorial inheritance, there are combinations of different factors, some genetic and others not genetic, that each by itself is not enough to cause the condition, but in combination you reach a threshold above. And with autism and with other multifactorial disorders, we can see different combinations of factors or genes in different families, and sometimes even within a single family. And for autism, some of these non-genetic factors that people think are involved are pre- and postnatal environmental exposures, epigenetics, the actual DNA sequence, but may affect gene expression and gene regulation, and then there are probably other unknown factors. Next slide. So what is the evidence then that genes are involved in autism? There's evidence from twin studies. That is that identical or monozygotic twins have a concordance for an autism phenotype that share all of their DNA and their genes. One would expect that if this was completely genetic, that the concordance would be 100%. The fact that it's less says there are other non-genetic factors, but it's still very high for a complex trait. For example, if one looked at obesity or adult cardiovascular disease, you might see concordance in 10 recessive pattern. You would expect that there would be a 1 in 4 or 25% concordance. And again, the fact that it's less suggests that not all of the same genetic factors are involved in siblings. Other evidence that genes are involved is familial aggregation. I think we all know now that if a family has had a child diagnosed with autism, it is greater than the population risk in general. It's been estimated at 8 to 10% after one affected child, although more recent studies suggest that it may be higher than 20 or even 30%, partly dependent on sex. And we also know that the recurrence risk is sex dependent, and this again is a feature that we see in multifactorial. We know that there are single gene disorders where autism spectrum disorders are a part of it and may be common. And in these conditions, it points to the importance of genetics, but also these disorders might give us clues about what might be going on in idiopathic autism. And I've just listed some of the disorders here. We'll And then finally, we know that chromosomal disorders and what we call copy number variants, CNVs, which is a term that people use frequently now to describe smaller genomic rearrangements, extra bits or missing bits of our chromosome material. And we know that these can produce autism. So in trisomy 21 or Down syndrome, and that the most common specific chromosomal change found in autism when people were doing regular chromosome testing was a duplication on chromosome 15, typically inherited from the mother, and this has been, this may cause between 1 and 3% of all autism. Leap in our ability to find underlying genetic and chromosomal etiologies in autism has been with a new technique that allows one to find smaller genomic rearrangements that can't be seen on the regular chromosome test. This is called microarray. The most common finding found in about 1% the 4-to-1 or higher male-to-female ratio that we see in idiopathic or isolated autism. And it is in this group where you find an even higher association with chromosomal abnormalities. Next slide. I also want to emphasize again the importance of new mutations. So new or de novo number variations are found in up to 10% of cases. Researchers have also found new changes in one copy of a dosage-sensitive gene where there is a single base or a couple of base mutation, and these are found in 1 to 2% of cases. The gene changes in particular are associated with older fathers, has provided very elegant evidence that the rate of mutations in male sperm doubles every 16.5 years. The rate is still low, so men in their 40s shouldn't get worried and completely stop having children, but this does explain some of the reason for perhaps new mutations and maybe even over the last 10 to 20 years as more slightly older couples are having children. The reason that you see more mutations of single bases in men is because sperm undergo many, many DNA replications in the lifetime of an adult male, and so these mutations can accumulate. Women maladies as they get older, but the DNA in egg cells only undergo 2 divisions, in a woman's reproductive years. Next slide. So before we talk about testing, I think it's important to ask the question, because I think I certainly get asked sometimes by families or other healthcare providers, right now in many or in most cases it's not going to result in treatment. It certainly in virtually all cases is not going to result in a cure. So what's the value of it? Well, one is that even though it may not sound like much, but it prevents additional unnecessary testing. Many families and many healthcare providers want to keep looking until they find a cause. This can also be very expensive, and funds that families might use for therapies and other purposes. So it prevents, if you know the cause, doing further testing. It may relieve some parental guilt. So parents may feel that, well, it was that one glass of wine that the mother took during pregnancy, or the time that the baby fell out of the crib when they were 2 months old. So you find a genetic change that is inherited from a parent, that could bring on other guilt because the parent unknowingly may have passed it on. So these are issues that we deal with in counseling. If we find a genetic or chromosome change that we have some medical information about, like Down syndrome or some of the more common CNVS, we may help predict future child will function as an adult. We may be able to tailor specific interventions. We hope, but it's not always true, but we hope that we can provide better guidance concerning recurrence risks if the family wants to have more children. For example, if the family has a new mutation that is not carried by either parent, then the risk of having another affected child may. And very rarely, but we don't want to miss these, it may permit specific medical therapies, vitamins, diet, medication that may improve the outcome. These are rare, and I always tell the family I don't want to hold out a lot of hope, but rarely we may find these, and certainly we don't want to miss it. Next slide. So now I'd like to go through what is recommended Academy of Pediatrics, by the Committee on Neurogenetics of the ATN, as well as other organizations. And it's really pretty simple right now. So a chromosomal microarray analysis is recommended. Doing a blood karyotype or a regular chromosome test is not recommended anymore unless there are very specific indications. If you see a Down syndrome, you may want to just order the chromosome test because it's a lot cheaper. But in cases where we even think we have a pretty good suspicion about what a disorder might be, we may be wrong, and you get good screening for all different known disorders as well as for some very rare unique ones on the array test. The other test that is recommended is in parentheses because some groups and organizations recommend it in boys and girls, some in boys with girls only if there is a high index of suspicion. I would recommend it in both. It's relatively cheap and has implications in terms of genetic counseling for the family because of the pattern of inheritance. And so I recommend it in all disability. Other testing that would normally be ordered by the pediatrician or developmental specialist or neurologist would be hearing screening as well as a lead level. Next slide. Second-tier testing, which we'll talk about a bit as well. PTEN gene sequencing, this is a single gene disorder that can be associated with macrocephaly and large. And then additional specific gene testing with other features, seizures, language regression, microcephaly, and this might be the point where one would consider referral to either a neurologist or a geneticist, depending on the institution. And then at the very end, I'm going to talk about what's the next tsunami coming through, which would be whole exome. I think today there is good data that metabolic screening is of very limited value without other indications, including for mitochondrial disease. I'd be happy to discuss this if there are specific questions, and I know there's been concerns about mitochondrial disease, but in my experience and in most of the literature published in small, not huge studies, present with typical autism without other features. EEG is not indicated routinely unless there's a suspicion for seizures. And while one may find some interesting variants or subtle anomalies on MRI, it really has minimal clinical diagnostic value unless there are other indications, such as an abnormal neuroexam, seizures, small head size, things like that. It's important to remember that follow-up is needed when you find a chromosome imbalance. This would include follow-up genetic testing studies, evaluation of the parents, often with a desire to see whether the parent has the same chromosomal abnormality, although there may be a lot of difficulty in getting this paid for. And then slide. So just to say a little more or a lot more about the array, as I mentioned, it's replaced the regular chromosome test in almost all cases. The list price cost is probably in the $2,000 to $2,500 range. Some insurance companies pay for this routinely, others do not, and I will say our genetic counselors spend a lot of time writing letters back and forth to ensure. The test compares a test or patient sample against a reference sample. So it takes a test and a reference sample together and compares them against bits of DNA that are attached to a slide or a chip and go up and down every chromosome. And if the test and the reference sample have the same amount the test and the reference are the same, the dosage is the same, you would see yellow here. If you have less of the test, it comes up redder. And if you have more of the test, a duplication, it comes up greener. And there are software that will go over every little bit and spot on these slides, and there may be hundreds of thousands to more than 1 million of these little spots, and analyze Next slide. This can be kind of complicated. The way I like to explain it to families, and I think nowadays I usually say a chapter book because nobody knows what a printed encyclopedia is like anymore. They're all online. But if you thought about your genome as a 24-volume encyclopedia or a large book with 24 chapters, chromosome, and there'd be separate chapters for the X and the Y chromosomes. Next slide. If you did a regular chromosome test, you could see a volume or half a volume extra and missing. That's the resolution when you look under the microscope. With the new array test, we can see pages, paragraphs, sometimes even complex sentences, and see much finer, smaller bits and pieces. But you still can't see a typo, which would be the equivalent of a single base mutation. With the next generation sequencing we'll talk about at the end, you can see typos, missing or added letters, and you, in theory, could pick one typo out of the 3 billion letters that are our genome. Next slide. The first is they won't detect regions that aren't represented on the chip. So they're very repetitive regions in our DNA that are not on there. And the older array tests back in 2006, 2008 were targeted and didn't cover every bit and piece of the chromosome. So if someone had a very early array, they might have an abnormality that you could pick up on a new array. Mentioned they won't detect single base mutations or very small, what we call indels, insertions or deletions of just a few bases. They won't detect balanced rearrangements. So the whole method is based on dosage. And it turns out that chromosome rearrangements, where two pieces on two different chromosomes swap places, but all of the information and genes are 1 in 500 healthy individuals. Doesn't hurt the individual, but they have a chance to have a child with an unbalanced rearrangement, and we see this in couples with infertility or multiple miscarriages. So in those situations, a chromosome test where you can see balances, but they're in the wrong place may be helpful. But in autism, intellectual disability, arrays will not detect very low-level mosaicism. So you may know that some cases of Down syndrome have 46 chromosomes with a normal chromosome complement and then 47 plus 21. The array should be able to detect 50% mosaicism, probably 30%. When you get down in the 5 and 10%, you may many cells. Labs may request parental follow-up, which again may be hard to pay for, and then different labs may use different reporting criteria. Some labs will report any abnormality they find. Other labs set a size cutoff so that if a genomic rearrangement is very small, they may not report it at all. Next slide. Even more can be difficult to interpret. Just like we all differ from each other and vary in single base changes, all of us have copy number variation. Some of these are very common, and some of these are unique in a family and have no medical significance whatsoever. We call these benign CNVs. But until you know it's benign, if it's unique in a family, you may not know what to do with it. It is this group that are the most troubling for us, and where we sometimes like to look at parents to see whether they may have the same thing or not. Next slide. So again, if we now look at arrays in autism, the first paper came out in 2007 showing a 10% de novo copy number variant rate in autism. There have been, look, the second bullet from the bottom, a very large study that reviewed 33 other studies with almost 22,000 patients, found a yield of 15 to 20% of a copy number variant that was likely or probably pathogenic in developmental delay, autism, or multiple congenital anomalies. There are two very recent research studies probably implicate additional genes. They are not changing the way I think that clinical array analysis is done presently, although this whole field is probably going to continue to evolve. Next slide. The way we assess the risk for CNVs, we think that they're likely to be damaging or cause disease if they overlap a known disease region, if they're large, if they have lots of genes in them, and we think in general that deletions, missing pieces, are worse than extra copies. Typically, benign variants are inherited from a healthy parent. We find them in other healthy individuals. There are not many genes in them, and in general, I never say never anymore, and I think of a case I had clinically within the last couple of years of a 10 megabase, 10 million base pair duplication in a child with autism that was called out as very likely pathogenic. I mean, that's huge. You could see it on regular chromosomes. When we did the dad, he had this exact same thing and was perfectly normal. So that was in a region that was well tolerated. May cause autism, intellectual disability, epilepsy, mental health disorders, learning disabilities, and different people in the family may have different clinical features. In most cases, or all cases really, we don't understand the reason for this. Most likely it involves other modifier genes or some environmental factors. And as I mentioned before, the early And so it's important, if you're looking for a cause, if the array was done 5 years ago, to repeat with the newest array. And there are certainly some older children, teenagers here that we see who've never had an array. And one should really think about, if you're seeing them in a medical management clinic, whether that child or family would benefit from. A little bit about Fragile X syndrome, which is the other first-tier test that's recommended. The incidence is about 1 in 4,000 in the general population. It's the most common inherited cause of intellectual disability. The carrier rate in females which carry a premutation is about 1 in 260, and 1 in 800 males. This gene, but the disease is associated with expansion of a triplet repeat at the 5', the beginning of the gene. It's not coding, but it affects the expression of the gene. And basically, all individuals, all males with Fragile X, inherit it from the mother, who would be typically a premutation carrier. So and it accounts for about 2% of all children with intellectual disability and 1/3 of all X-linked cases. Next slide. These are some of the features that we see in males. At our center, most males with Fragile X are diagnosed as young infants because of hypotonia or delays on your center, and certainly there's some unique behavioral phenotypes in these individuals. And there is an increased risk of seizures, and it's very important because of risk to other members of the family to make this diagnosis. Next slide. These are some pictures of individuals with Fragile X, 3 boys, an older male. We typically talk about are that boys with Fragile X may have a long face, large ears. You can kind of see that in the males, and this becomes more prominent as they get older. They may have a prominent jaw and chin, but it's not seen certainly in 100%. Girls with Fragile X syndrome may look completely normal and typically have no dysmorphic feature. Fragile X and autism, if you looked at an autism population, statistics would say 3 to 6% of children would have Fragile X. About 25% of males with Fragile X meet criteria for classic autism, and more than 50% would test on the spectrum. Girls that have the full mutation, about 1/3 of them will have intellectual disability, up to 10% with autism, and 1/3 on the spectrum. And again, this has to do with some of the unusual genetics about Fragile X and patterns of X inactivation in the brain in females. It's interesting that there is some data that girls that carry the premutation, so not the normal range, slightly increased number of these triplet repeats, this might be a susceptibility locus for all. Next. Second-tier testing. Next. I want to say a couple of words about Rett syndrome. This is X-linked dominant, usually male lethal. So in this case, girls with 2 X's have 1 normal X, 1 X with a mutation in the Rett gene, and you have to have some normal amount of this protein in the brain, or you typically wouldn't. They die before birth. Seen in about 1 in 10,000 females. It results from mutations in a gene called MECP2. You can see some deletions or duplications of the gene. And the clinical features in a classic case, the girls typically start off normal with regression between 6 and 18 months of age. They then lose speech and seizures, and they have typical stereotypic hand washing or hand movements that when you see them in a classic case, you can't forget them. Next slide. So on the right here is a girl. It's not a video, but you see she's holding her hand at the midline. She will ring and do washing motions with these. I've seen other girls that will move their hands back. with severe neonatal brain dysfunction and die within the first few months of life. And then there are rare females reported without other features of Rett syndrome that may just present with intellectual disability or autism. And this is why some groups recommend as a second-tier test doing MECP2 sequencing in all girls and maybe even males. A few words about PTEN. So PTEN is a tumor suppressor, and it's a lipid phosphatase. It was first described as part of an adult cancer syndrome called Cowden syndrome, and it is an autosomal dominant disorder. So one mutation in the PTEN gene, other copy normal, and you have Cowden syndrome. It was later found in something called BR, mild intellectual disability, lipomas, other hamartomas, and in boys, in the second half of the first decade, they will often get speckling on their penis. It was then seen that in some Cowden families with PTEN mutations, there were children with large heads and autism, but without anything else. And so in 2005, Butler and colleagues sequenced several patients with autism and macrocephaly, families where there was no Cowden syndrome. We and others have looked at a series of patients, and it's now felt that maybe 5 to 10% of children with macrocephaly, large head greater than 2 standard deviations, and ASD or developmental delay will have a PTEN mutation. Most of these children, the head is large at birth, and it's 2 to 4 to even greater standard. And the phenotypes can be very variable even within a family. So you can have an affected child, another child may just have a large head in PTEN and not have any intellectual or autism problems. Next slide. These are a couple of pictures from the internet of children with PTEN. They're not typically dysmorphic, but have this very large head, usually from birth. Important to recognize, because if it is inherited from a parent, the recurrence risk to pass it, the gene mutation on, is 50%. A parent may pass it on and have a child with just a large head with no autism, but certainly there is a higher risk. Because of the increased cancer risk, it's also important to identify affected children and parents to institute medical surveillance. This is just a list of when to maybe think about a genetics referral. I'm not going to go through everything. And in some cases, depending on your institution, you may make a neurology referral versus genetics. But there's a long list here. And certainly, if there is a desire of the family, if there are 2 or more affected and they're interested in recurrence risk, making a genetic. So now I just want to finish a little bit with whole genome and whole exome sequencing. Next slide. So just a couple of facts again in a little more detail about our genes. So we have about 23,000 genes organized on all of our chromosomes. You may remember that only 2 to 3% of that material is the actual gene itself in all of our base. Genes are broken down into coding bits, which are the exons, separated by spacers, which are the introns, and there's a complex machinery in the cells that takes the DNA and makes it down into RNA, where the little introns, the red bits, are chopped out so that the final message that gets converted into protein is contiguous. Next slide. Next, enable rapid sequencing of large numbers of pieces of DNA all together, something that wasn't possible even 10 years ago. When we talk about whole genome sequencing, we're determining the sequence of most of the DNA content of all of our DNA, except for regions that are very repetitive, you can't do well. Whereas the exome regions, rather than all the stuff in between the genes and all of the introns. Next slide. So why would you do exome sequencing over the whole genome? It's about 1% of the genome, and today it's the part that's most likely to include interpretable mutations that result in clinical phenotypes. A nonsense mutation, so in the middle of a protein there's a mutation that stops the protein right in the middle, you pretty well think that's not going to be good. We know a lot about missense mutations that change the coding amino acid sequence. So if we get a change in the DNA, we're more likely to be able to understand if it's significant or not. The sequence exome, but the analysis costs are much lower, and it's the analysis, taking all of those little bits of DNA sequence and organizing them. It's like taking that whole 24-volume encyclopedia, chopping out every word, throwing it up in the air, and then asking someone to put it all back together again in the right order. It's much easier to do when you just look at the coding gene bits than when you look at everything. Some coding exons that for one reason or another are difficult to sequence or capture the way this is done today. Next slide. So how is this done today? Simply for exome sequencing, you take genomic DNA from a blood sample and you chop it up into little precise fragments. And then you hybridize those fragments with a commercial library that isn't in either parent, then it would be new, and it's pretty easy with the software and analysis tools to look for these kind of events more than to sort through the thousands of inherited changes that we all have to find those that might be causing problems. Most of the changes that were found in genes were found in a single individual, and these a change in the same gene in two or more unrelated individuals, it's so unlikely to happen by chance that it makes it, just finding it in two people, a new autism susceptibility gene. These kinds of new changes are what people are looking for mostly now when they're doing exome sequencing in autism, but they're still found in only a small number of families tested. Next slide. Many of these are probably inherited, and as I mentioned at the beginning, it's probably combinations of these, 5 to 10 per person plus environmental factors that lead to autism. Other variants may affect gene expression and regulation and don't occur in the coding part of the gene. And so we won't find these until we look at the whole genome. And if analyzing the exome is hard, analyzing the whole genome today both commercial and academic that offer it as a clinical test. I think in a disorder like autism, unless you're lucky enough to find one of these new mutations in a gene that's already been described or that has a very important role in brain development, interpretation is very hard. So I am not recommending it currently to families that I see with autism unless there's some whole exome sequencing on a child with autism, and you find a change, a mutation that you know could be problematic in the breast cancer, BRCA1 gene. Well, you can say, well, the child's 5 years old, he's a male. It doesn't matter. I don't need to report that. But he almost certainly inherited that change from one of his parents. And if he inherited it from his mother, and so when one thinks about, well, we don't give out results like this in children, these can apply to the whole family, and so there are a lot of issues that need to be dealt with. Next slide. And so, again, the American College of Medical Genetics and Genomics feels currently that whole genome or whole exome clinical sequencing should include pre- and post-test counseling. That we're all going to still need to deal with in the future. Thank you. And I guess I'll be happy to take some questions.
Great. Thanks very much.